Chapter 5, the Integumentary System. We're going to introduce the skin, or the integument system. The organs of the integumentary system include the skin and its accessory structures, which include your hair, your nails, glands, as well as blood vessels, muscles, and nerves. Functions of the integument system. First and foremost, our skin serves as a major protector. It's protecting us against dehydration, invasion of bacteria, and mechanical injury. So the next question is how? How does it protect us against dehydration, this bacterial invasion, and mechanical injury? If you go back to our tissue lab when we talked about our stratified squamous epithelium, you remember that we had a modification, a protein modification called keratin. And that was an important modification that hardens and waterproofs that uppermost layer of our skin to protect us. Then next is going to be temperature regulation. Now, during cold weather, blood vessels within the skin vasoconstrict and are held closer to the skeletal muscle. If you think about when it's cold out, look at your fingers. What color do they turn? More white. Why? Blood is being restricted from those areas and being diverted to the core to maintain internal body temperature. Now, heat is picked up by the blood, whereas in hot weather, blood vessels vasodilate and bring the heat closer to the surface of the skin. Combined with sweating, this cools the body. So if you think about exercising, what happens to your skin when you're exercising? You turn red. Your face, your arms, your chest, your back, your legs. What's happening? Your muscles, your skeletal muscles are doing work. They're converting energy to heat. So if you remember a law of thermodynamics that states energy can neither be created nor destroyed. So all that kinetic energy that's being created from your muscle work is transferred to your blood via heat. Now your blood has to cool somehow, so we vasodilate all the blood vessels to the largest organ in our body, which is our skin, bring it to the surface, allow it to cool ambiently with the air, and it's a really efficient way of cooling us, in combination with sweating, of course. We have a few more functions of the integument system. One is going to be sensory detection. So, we have sensory nerves, receptors for crude and light touch, pain, and temperature that allow us to interact with the environment around us. Another function is vitamin D3 synthesis. This is really important. It is a steroid present in the skin which is converted into vitamin D when exposed to UV light. That's why it's really important to get outside in the sun and really soak up those rays. The reason why is because in the kidneys, vitamin D is converted into a hormone, calcitriol. And calcitriol is responsible for managing bone growth and calcium absorption by the bones. So we're getting bone strength and bone rebuilding efficiently when we have high amounts of vitamin D. We're also going to excrete metabolic waste. So we say, well, how do we do that? The answer is sweat. Now, sweat is basically a dilute urine, and a lot of people here say, oh my God, it's urine that's coming out of my sweat pores, and the answer is yes. Think about if you wear a white t-shirt consistently to the gym, where you sweat, where you have the highest concentration of sweat glands under your arm, right? What color does that shirt turn over time? Yellowy and brown. Well, that's because we're dealing with dilute urine here. Let's look at a quick focus on homeostasis. The contributions of the integumentary system for all body systems. Skin and hair provide barriers that protect all internal organs from damaging agents in the external environment. Sweat glands and skin, blood vessels regulate body temperature needed for proper functioning of other body systems. You could go through each of these systems and see how they contribute to our other mastery systems. The next part of skin that we have to examine are the layers. So let's take some time and identify all these layers. So our first layer that we're going to see is going to be our cutaneous membrane. Now that cutaneous membrane is made up of two layers, one an epidermis, two dermis. Now again, when you talk about the terminology here, dermis meaning skin, epi meaning on top of. Now we have another layer that's not really part of the skin, but it is a subcutaneous layer. It's a layer below that cutaneous membrane, and it is called the hypodermis. So again, hypo, here's our Latin for below, meaning below the dermis. And this is where we're going to find a lot of our accessory structures. Let's use a picture to examine some of the structures of the skin. So we're going to start with our layers. First, we have our epidermis, right? So that is made up of our stratified squamous epithelium with our keratinized layer. Then we have our dermis. Now, the dermis is important because that dermis is made up of two layers, a papillary layer and a reticular layer. Now, the majority, if you look at this bracket here, the majority of the dermis is made up of this reticular. The papillary layer, and then you can see, look here, you can see almost where the arrangement and types of fibers change in this upper to lower layer of the dermis. Now, these projections, these papilla, these little finger-like projections, are going to increase surface area to form almost like an egg crate or zipper-like connection between the epidermis and the dermis. The last layer is right here. This is our subcutaneous layer. Look, there's a lot of fat located in here, a lot of protection, insulation, and energy stores. Now let's look at some of our accessory structures. We have a pore, so pore of a sweat gland, which brings us to our sweat gland right here. We have a really important muscle called the erector pili muscle, which is attached to every hair follicle. Now, attached also to the hair follicle is a sebaceous gland, okay? So we say, well, why do we have a sebaceous gland, an oil gland connected to a hair follicle? Why a muscle? Well, the sebaceous gland is a protective in nature. So every hair that we have coming through our skin is essentially a hole. That hole is a prime spot for bacteria to come in, invade, and try to attack us. So the way we protect it is by filling it with oil. Next, the erector pili muscle. This is a vestigial structure. It really serves no function anymore other than giving us goosebumps and making our hair stand up on end. But it was very important when we were in our early hominid stage. Reason being, remember, we were covered in hair. So what happened? When we were scared or we heard a creepy noise, this muscle would fire as a defense mechanism and make us into this huge fur ball. If you've ever seen a cat scared or dog with a lot of hair scared, when they get into that fight or flight response, they turn into a huge fur ball. So we had that mechanism. The next was temperature control. Again, when we were covered with hair, the way we helped regulate living outdoors, especially in the cold, was to fire this erector pili muscle, putting all the hair on our body straight on end, essentially making an air blanket. Our body would warm that little air blanket that was trapped between the hair and the skin and essentially keep us regulated. Another couple structures that we see are going to be lamellated corpuscle. This is known as Pacinian corpuscle. We'll talk about what exactly these do in conjunction with tactile corpuscle. We have some nerve fibers, and when we get into a more detailed picture, we'll see that there's a nerve plexus, and we see right here this nerve plexus is going to surround every hair follicle, and then again our sweat gland. We have our cutaneous plexus for arteries and veins, and some adipose or fat here. This is a histological view of the epidermis, and we're going to look at this again when we identify some of those major layers within the epidermis and dermis. So let's look really quickly. Here's our epidermis. It is this purple region, and above you can see the wispy keratin. Now we can move down into the dermis. We have our two regions, our papillary region, followed by our reticular region. We have our hair root with hair follicle, and you can see, look at this sebaceous gland filled with all these oil cells that are going to secrete that oil to lubricate that opening, that pore. Let's look at the epidermis into detail. Let's look at our four types of cells that we find here. First are keratinocytes, and as you probably guessed based on the name, they're responsible for making keratin. This is the protein that is responsible for giving the strength and flexibility for our skin. The next cell is a melanocyte. Now, their role is well known for skin pigmentation. They have this unique ability to produce and distribute melanin. So let's write melanin. The next cell are called Langerhans cells. These are known as intraepidermal macrophages. Now, these cells act as the outermost guard of the cutaneous immune system, and they're typically going to induce the first reactions against pathogens that are encountered by the skin. So these are strictly an immune function. The last cell we're going to look at are tactile epithelial cells. They're called Merkel cells. So these are oval-shaped mechanoreceptors, and they're essential for light touch sensation, and they're found specifically in our skin. So we could put it to type. Here's our keratinocytes, produce keratin, this tough fibrous protein that provides protection. Melanocytes produce the pigment melanin. Intraepidermal macrophages are involved in the immune response, and tactile epithelial cells function in the sensation of touch. Skin pigmentation. So as we said earlier, our melanin is produced by melanocytes in the stratum basals. Now we haven't talked about all the different layers yet, but as you probably inferred, Latin stratum means layer. Basal, as we've seen before, is that lowest most layer or basement membrane, right? So basement layer, so lowermost layer. Now we see that this cell has these appendages, these little arms, that is an arm making stuff. So the cell is programmed to make stuff. It's making these little melanin granules that will be released to govern skin pigmentation. Let's get into the 4 layers of the skin. Now remember that we have 2 types of skin. We have thin skin and thick skin. Now, thin skin is going to be found in the majority of our body and is comprised of 4 layers, 4 stratums. Thick skin is the soles of our feet and palms of our hand. And because it's thick, it's going to have an additional 5th layer. So we're going to cover all 5 layers and then exclude that 5th one from thickened skin. Now we can also call thin skin as hairy skin and thick skin as hairless. So we'll say thin skin is hairy while thick skin is hairless. And this is pretty easy to see, right? Look at your palms, look at the bottom of your feet. Is there hair there? And your answer should be no. Right? And look all over your arms or chest or back or legs and you say, well, this is thin skin. Is there hair there? And the answer is most likely yes. Now here's our 4 layers. First we have the stratum corneum. And again, corneum means corny or horny. When you think about what animal has a horn, like a rhinoceros, and what is that horn made out of, it is going to be keratin. So we're working from top to bottom, superficial to deep. The next is the stratum lucidum. This is that clear layer that's only present in skin. So lucid means clear. Next is going to be the stratum granulosum. And as you've probably guessed, this is going to give a grainy appearance. Next is the stratum spinosum. So this gives this spiny appearance. And then lastly, the stratum basal, which is the bottommost 1 cell layer thick, mitotically active, 1 cell layer thick, mitotically active layer, mitotically active. Now I've come up with a mnemonic to help you work from, again, superficial to deep. This may work for some students, it may not work for others. You're more than welcome to come up with your own, whatever helps you learn these layers. So starting from the superficial most layer, the stratum corneum, and working lucidum, granulosum, spinosum, basal, we write that classy ladies get sexy boys. Okay, so that's our mnemonic. Classy ladies get sexy boys. Now, let's look at these 5 layers. It's going to replace itself every 30 to 35 days. So if you get a little cut, we know that within a month or so, that cut will be completely replaced with new skin. Now again, where is and how is it replacing? right here, the stratum basal. This is what's mitotically active. Let's look at a histological view of these 5 layers. So we can look at a picture and then we'll look at actual microscope slide. So on the left, we have our dead keratinocytes. So this is our stratum corneum. Then we get this clear layer here, and we're going to outline exactly what's in these layers in a little bit for the stratum lucidum. So now, because we see the stratum lucidum right off the bat, you should be thinking thick skin. Then we get these speckled, granulated cells right here. Okay, and that's our stratum granulosum. And this spiny area from here to here is the stratum spinosum. This one cell layer thick at the bottom is our stratum basal. This is the mitotically active. And you can see, I'm going to erase right here. You can see that there is a cell undergoing cell division. This is telophase down at the bottom over here. We also have a Merkel's disc. So let's erase this section. We have a Merkel's disc right here, tactile for fine touch receptor, as well as a Langerhans cell, so an intraepidermal macrophage. So we get a view of everything that's located within these layers. Now let's look at this histologically from a slide perspective. So if you go on my YouTube channel, Anatomy and Physiology Crack, you'll see the differences between thick skin and thin skin. Again, this is thick skin. How do we know? Everything above here, keratin, so that's our stratum corneum. Here's that nice red clear layer, stratum lucidum. Speckled black grainy layer, granulosum. Here is our spiny layer, spinosum, and at the bottom, here, one cell layer thick, stratum basal. Now, one other thing to look at, all these little projections that move into the spinosum are called dermal papillae. One other thing to see here, let's erase this line I drew because I drew over another structure, is a corpuscle of touch. You can see this nice big corpuscle of touch right here in the dermal papillae. Let's get some detail into the epidermis, these 5 layers. The stratum corneum, remember we said that this is our keratin layer. So we get about 15 to 30 rows of dead cells. Keratin provides our water resistance or waterproofing, but we do allow some water to perspire or evaporate through our skin, about 500 mils of insensible perspiration. So that gives us 2 types of perspiration, insensible versus sensible. Now the sensible perspiration can vary, right? And you say, well, why can this vary? Well, it depends how much you're exercising. So sensible perspiration is the actual water that you see dripping off of your face, your arms, your body when you're exercising for an extended period of time. Now, the insensible is what you don't see. So about half a liter of that coming through our skin. The next is our stratum lucidum. Again, palms and soles of our feet. 3 to 4 layers of thick dead cells packed with keratin. Again, it gives that clear appearance. Our stratum granulosum, that grainy layer, is the middle layer, 3 to 5 rows here. And the cells here are going to produce a lot of keratin and stop dividing. There is no mitosis going on here. Our next layer, 4th layer, is the stratum spinosum, that spiny looking cellular layer. 8 to 10 rows of cells. Some here can divide, some cells here can divide. We have our Langerhans cells, our macrophages that we saw previously, which is protecting against microorganisms, as well as superficial skin cancers. Our lowest layer, our stratum basal, is one single layer of mitotically active cells that replace all the cell layers superficially or above it. Our epidermal ridges are present here, extend into the dermal papillae, creating our fingerprints. I'm going to talk about fingerprints in a little bit. We're also going to find our Merkel cells, receptors for light touch, and our melanocytes, which is going to stimulate pigmentation in our skin. So as a review, our types of skin include thin skin, which is also known as hairy, covers all body regions except for palms, palmar surfaces, and digits and soles. Our thick or hairless skin, which covers the palms and palmar surfaces, and surface of digits and soles. Now that we have finished the epidermis, it's time to look at the dermis. The dermis is composed of connective tissue containing collagen and elastic fibers, so support and elasticity. So if you pinch your skin and it elastically recoils back down, snaps back down relatively quickly, this means one, you are well hydrated, and two, those elastic fibers are still intact. It contains two regions. The papillary region, which as we said before, which is much smaller and takes up less percentage of the dermis, lies just below the epidermis. And the reticular region, which is going to consist primarily of dense, irregular connective tissue. Remember from our tissues lecture, these bundles of collagen fibers running in irregular or crosshatch motif. This is one of my favorite pictures, and I'll tell you why. I like how they pull up here the epidermis. So you really get to see the way the spinosum and the dermal papillae interlock, zipper together to form a high surface area, multi-point attachment for those desmosomes and hemidesmosomes to link and lock this upper layer into the middle layer. So it's one of my favorite pictures, again, showing you that egg crate shape, or if you take your fingers and interlace them, you can mimic how these fingers, these papillae, interlock, increasing surface area. And again, structures we've seen before, the erector pili, sebaceous gland. Here's a corpuscle of touch. We have a deep lamellated, so corpuscle that's lamellated means covered in fat. We have a hair root plexus here. Our reticular region, our papillary region up here, reticular region down here, and our subcu or hypodermis down here. Fingerprints. Let's talk about fingerprints for a little bit. We already established where they are made and set up, but why do no two people have the same fingerprints? And the answer is pretty simple, because our fingerprints are established in the uterus. So how are they established? Even with monozygotic twins that are developing in the same womb. Well, by tactile touch. So as that baby is moving around, as that fetus is moving around, everything it touches is forming these dermal or epidermal ridges, forming our fingerprints. That's why no baby, not even monozygotic twins, have the same tactile touch within the womb. They're pushing against each other, pushing against the placenta. So no two feedbacks are going to be the same. Now, the next thing to look at is that we do have sweat pores located in here. And the reason why is we're allowing for heat transfer or loss of heat. Now, you can see this, right? And even excreting waste. So if you put your hand on a black table for a few seconds, you lift it up, you see that water or that wet handprint that remains, that is from your sweat pores. Now, why do we have these epidermal ridges? What's the purpose of them? What's the point of them? Well, they give us an increase in surface area to allow us to grab and hold on to things, right? If we didn't have these, you try to grab something, it would just slip right through. Now, when and how can this change? Water. Let's put ourselves into a pool of water. What happens over time? We increase the surface area by wrinkling our fingers. This is an adaptation. This is our body saying, hey, we're not supposed to be in this type of environment. If I'm going to live here forever, I need to be able to grab things. So it wrinkles the skin, increasing surface area on top of these epidermal ridges to maximize the ability for us to grab things. So table 5.2 summarizes the papillary and reticular regions of the dermis. So you can read through this. We'll read through some of the details. So the papillary is the superficial portion of the dermis, about 1/5, consists of areolar connective tissue. And then we get these thin collagen and fine elastic fibers. They're going to contain the dermal ridges that house blood capillaries, corpuscles of touch, and free nerve endings. Our reticular region is the deeper portion, consists about 4/5, and it's going to have our dense, irregular connective tissue. These are our bundles of thick collagen fibers. We get some coarse elastic fibers here. The spaces between fibers contain some adipose cells, hair follicles, nerves, sebaceous glands, and sudoriferous glands. The last layer we have to talk about is our subcutaneous layer. It's also known as the hypodermis. It attaches to the skin, to the underlying tissues of the organ. So yes, the subcutaneous layer is not technically part of the skin. It is an attachment layer, attaching the skin, the dermis and the epidermis, to the underlying tissues and organs. The next thing we have to talk about are our sensory receptors. So what's detecting stimuli? Touch. The skin contains different types of sensory receptors, and they're found in two different layers. They're found superficially. These are type 1 cutaneous mechanoreceptors. So mechanoreceptors detect stretch. And free nerve endings, again, why do we differentiate free from encapsulated? Free will not have fat, so no fat surrounding it, where our lamellated corpuscles down here deep are covered with fat. Okay, this is an insulator, so it's insulating. Okay, so aside from our free nerve endings, we get corpuscles of touch and our hair root plexus. So you can test your hair root plexus. Blow on your arm. Blow on your arm. You can feel the hair move on your arm because of those hair root plexuses. So we can put it to picture. So again, a typical repeat. We outlined this for you before, but let's look at a couple things. We have a melanocyte over here, right? You can see those arms with those melanin granules in there. Merkel's disc, so again, a fine touch or tactile receptor here. Our keratinocytes here, our granulocytes, lucidum, and dead keratinocytes, cornea.